everyone and welcome to The Dentist Hub. If you haven't subscribed to my channel yet, don't forget to subscribe and also follow my page on Instagram. Links will be given in the description box below. So in today's video, we'll be learning about the culture media and its various types. And it's also an important question asked in microbiology, so make sure you watch the video till end. Culture media is used to grow organisms from infected material to identify the causative agent. After identification, it is also used for antibiotic susceptibility. The composition of culture media. It consists of water, which acts as a source of hydrogen and oxygen, agar, which is prepared from seaweed and acts as a solidifying agent, electrolytes like sodium chloride or other electrolytes, peptone, which is a complex mixture of partially digested protein, meat extract that contains protein degradation products, and blood serum for enriching the culture media. The culture media is broadly divided into the simple and the complex media. The complex media are further divided into special and synthetic media. The complex media are all other media other than simple media. These have added ingredients in order to bring out certain properties. The synthetic media are prepared from pure chemicals. The exact composition of media is known. Now let us discuss in detail about the simple and the special culture media. The examples of simple media are the nutrient broth, which is prepared from peptone water and 1% meat extract. The glucose broth is prepared from nutrient broth and glucose 0.5%. The nutrient agar is prepared from the nutrient broth, where 2 to 3% of agar is added. This is the simplest and routinely used culture media. Coming to the special media, the different types are enriched media, enrichment media, selective media, differential media, indicator media, transport media, sugar and anaerobic media. Now let us discuss in detail about each one of these. The enriched media. In enriched media, the basal medium is added with some nutrients such as blood, serum or egg. Some of the examples of enriched media are blood agar, where sheep blood 5 to 10% is added to the nutrient agar. Hemolysis is observed in case of streptococcus. Different types of hemolysis is used to differentiate between various gram-positive bacteria. Thus, blood agar also acts as differential media. The chocolate agar. It is prepared by heating the blood agar for 2 hours at 55 degrees centigrade. It is used for isolation of Neisseria species and Haemophilus influenzae. The Loeffler serum slope. In this media, horse serum is added to enrich it. It is used for the isolation of Corynebacterium diphtheriae. Coming to the enrichment media. Here, substances are incorporated into the liquid media to show stimulating effect on bacteria to be grown or inhibits its competitors. Thus, there is an increase in number of wanted bacteria. It is useful in culture of feces. The examples of enrichment media are the tetrathionate broth, where tetrathionate is added to the nutrient broth. It inhibits coliform bacteria and allows paratyphoid bacteria to grow. The selenite F broth. Here, sodium selenate is added to the nutrient broth and it helps in culture of Salmonella and Shigella. Other example includes alkaline peptone water, whose pH is 9. It is used in culture of Vibrio. The selective media. It is a solid media and it is used to isolate particular bacteria from specimen where mixed bacterial flora is expected. The examples include the bile salt agar, which is used to isolate Vibrio cholerae, the deoxycholate citrate agar, which is used to isolate the enteric bacilli. The differential media. It helps in distinguishing differential characteristics. Examples include the McConkey agar, which consists of peptone, lactose, agar, sodium taurocolate, and neutral red indicator. The lactose fermenters on the McConkey agar form pink-colored colonies, whereas the non-lactose fermenters form pale colonies. The indicator media. This media contains indicators. They change color when bacteria grows on them. McConkey agar is also an example of indicator media. Other examples include the Wilson-Blair medium, where it shows black colonies when Salmonella typhi bacteria is grown. The transport media. It is used for delicate organisms that may not survive the time taken for transit. The examples include the Stewart transport medium. It is a non-nutrient soft agar gel. The reducing agent present in it prevents oxidation and the charcoal neutralizes bacterial inhibitors. It is used to transport gonococci. The sugar media. It consists of 1% sugar in peptone water along with an indicator. It helps in identification of bacteria. Glucose, lactose, sucrose, and mannitol are routinely employed for fermentation tests. A small tube called the Durham's tube is kept inverted in the larger sugar tube to detect gas production. The anaerobic media. It is used for isolation of anaerobic bacteria. The examples include the thioglycolate broth and the cooked meat broth, where pre-digested cooked meat is added to the nutrient broth. Here's a quick list of various culture media that we have discussed, along with the bacteria that are isolated in them. Thank you for watching. Do like the video if you found it informative, share it with your friends, and subscribe to the Dentist Hub for more updates.